Hello everyone, welcome to another video from Shomu's Biology. In this series of lectures, we're talking about post-transcriptional modification to the eukaryotic mRNA. In this particular lecture, we're going to talk about RNA editing. What is RNA editing, how it is done, why it is done, and the physiological significance of RNA editing and mechanism of RNA editing and steps of RNA editing process in details. Now, in this series of lectures, we are talking about all the post-transcriptional modification in eukaryotic mRNA. Means right after the transcription of eukaryotic mRNA, it is premature mRNA, need to be modified. It's modified based on the 5' capping, 3' polyadenylation, splicing, and these are the three steps that generally done to make it mature enough. But RNA editing does not come under that roof of RNA processing in eukaryotes. But still RNA editing occurs right after the RNA transcription. So that's why we call it post-transcriptional modification. But RNA editing is not a complementary part of post-transcriptional RNA processing. Remember that in your mind. RNA editing is a process of modifying the RNA sequence in such a way which alters the RNA codons from the DNA information stored in the DNA based on either modifying the existing nucleotides in the DNA or either modifying the nucleotide existence in the RNA or inserting or deleting nucleotides from the existing RNA. So RNA editing is a process which is post-transcriptional modification but not under the roof of post-transcriptional mandatory mRNA processing. Keep this in your mind. So in RNA editing, it is not only common in animals, it is seen in plants, it is seen in animals, it is seen in fungi, it is also found in protists. It is there in all the different types of organisms that are listed in here. So that is RNA editing in the very first step. Now we need to understand why RNA editing is very important. So I always look at all this process from a purpose colored spectacles. That is the functional impact of RNA editing. What is the functional impact of RNA editing? Why RNA editing is so important? That is something that we need to understand first. The functional impact of RNA editing is something important because whatever information that is present in the DNA, let's say this is the DNA. So let's say one of this DNA strand is acting as a template strand for RNA. So this is the template strand from RNA. So whatever the template has, the RNA will have complementary structure to that. So this is mRNA. But this structure should be complementary, right? But if this RNA is edited, then it will have some loss of complementarity. Means the RNA will be modified in such a way, some portion of the nucleotide of the RNA can be modified to totally different nucleotide. So while they read in the translation, I'll take a different color, take a red color. So in the process of translation or protein synthesis, the protein produced from here in the unedited form of the mRNA, remember this is unedited form of the RNA, and this is also mRNA, but this mRNA is edited form of the mRNA. Okay, so unedited form of the mRNA produces a protein, let's say that is X, but the edited form of the mRNA will produce a protein that is, let's say, Y. Completely different proteins can be made, and that is the beauty of RNA editing. RNA editing alters the translation potential or expression potential of existing mRNA. So from the same DNA, we can produce multiple varieties of mRNA, which can give rise to multiple varieties of proteins in the cell. That is the beauty of mRNA editing. Changes the expression potential of mRNA in eukaryotes, in animals, in plants, in protists, in not only eukaryotes but also in all these type of examples that I already told you. So that is the importance of mRNA. And what is the physiological function? And I mean biological importance. We've seen this mRNA editing leads to various kinds of protein production, and the chances of variety of proteins can be produced by the cell increases. As a result of which it has very important prevalence in neurological disorders and also developmental disorders. It is also involved in the RNA virus mediated damages to the DNA. So these are all important biological significance of RNA editing. Okay, keep this in your mind. Now I'll talk about the types of RNA editing. What are the different types of RNA editing? So the types can be either modification of the existing nucleotide, and the example of such modification is that the adenine can be modified to inosine. This kind of modification can be seen. Cytosine can be modified to uracil. So these are examples of modification that can be found in RNA editing. And there is another set of RNA editing type that is not modification, that is either insertion or deletion. Means a particular nucleotide can be inserted, a particular nucleotide can be deleted from the existing mRNA. And also a third kind is there, that is site specific modification. Means any particular nucleotide in the whole chain of mRNA can be selectively modified by RNA editing. So these are the three different steps of RNA editing that we all know. Either modification of the existing nucleotide or insertion or deletion of new nucleotides can be done. Now what is the mechanism of RNA editing? We'll see the process of RNA editing in action, the steps in details in the next slide. But here we'll talk about the mechanism of RNA editing. The mechanism of RNA editing is fairly simple, and that is we have specific enzymes activating in the process of RNA editing. Those enzymes will act on locations of the RNA which needs to be edited. So those protein complexes will bind to specific region of the mRNA. And once the protein complex bind to the specific locations of the mRNA, then they will recruit other enzymes which will ultimately trigger the change to the RNA. Now the change can be a single nucleotide modification or nucleotide insertion or nucleotide deletion. Okay, so these are all the mechanisms of RNA editing. And also remember one more thing is that this RNA editing's direction means basically what happens is that the RNA editing can also be done in a guide RNA specific manner. Let's say this is the RNA which needs to be edited. So what we have in our hand is a guide RNA, a small stretch of the RNA which is already produced in the cell. The small stretch of the RNA known as guide mRNA or guide RNA binds to it. This is guide RNA. And in a particular location where they need to add or delete any nucleotide, then they'll cleave that part of the RNA and insert a new nucleotide or cleaves the existing nucleotide out and then rejoin the RNA. In that way, they will modify the existing mRNA. This is how the guide RNA mediated insertion or deletion can be done. On the other hand, there are several enzymes involved in the process of RNA editing to modify the existing nucleotide, that is A to I, C to U. So here comes the third important part, that is the enzymes. What kind of enzymes are involved in this process? So one enzyme is adenosine deaminase, known as ADAR as well. Adenosine deaminase editing machinery for the RNA dependent machinery, basically known as ADAR enzyme, ADAR. A simple terms, adenosine deaminase. So adenosine deaminase that edits the existing RNA. Now this enzyme adenosine deaminase, as the name suggests, will modify the adenosine residue, modify A, and will modify A adenosine. So what kind of modification? Deamination. So basically NH2 is hydrolyzed and cut out of the adenosine to convert adenosine into inosine. And the moment adenosine is converted to inosine, now this inosine is not a normal base that is found in the RNA, but once the inosine is present there, during translation, the inosine will be read as guanosine. So earlier it was there as adenine, but now adenosine should be there, but now as the I is inserted, it is read as guanosine or G. So that is treated like G. So during translation, different codon is read. That is one issue with this process. And cytosine to uracil again, deamination. Or cytosine to uracil can also be done with methylation. Cytosine, if methylated in 5 carbon position or 5th carbon position known as 5-methyl cytosine, then the cytosine will behave like uracil. The structure of cytosine will turn into uracil like behavior if the 5' carbon of the cytosine is modified with methyl group or attached with methyl group. Okay. So these are the enzymes that are involved. 5-methyl cytosine modification, an ADAR enzyme that is here. Apart from that, there is another enzyme that is involved in this process known as ApoBec. Known as ApoBec, C at the end, ApoBec, apolipoprotein mRNA editing complex. Apolipoprotein mRNA editing complex. This ApoBec is another enzyme set which modifies the apolipoprotein, which is a very important protein in our eukaryotes, in humans particularly, if you talk in mammals. This apolipoprotein modified to different form of the protein's length. The length of the protein is altered. One is bigger one, another one is a truncated form. Both of these forms have different functions to play in the body. Okay, that is how it's done. These are the type of enzymes that are actually involved in the process of mRNA editing. Okay, so these are all the enzymes. The types we have seen, either insertion, deletion, that can be done with guide RNA mediated editing, or modifications can be done, that is A to I, C to U can be done. Enzymes involved ADAR and ApoBec that are involved in the process, and we have already talked about the functional role that RNA editing plays. The functional role of RNA editing, as I mentioned earlier, is very important, that it is related to the neurological disorders, the prevalence in the neurological disorders, and also involved in the RNA mediated or RNA virus mediated process and changes. And the more important thing of RNA editing is that to give enormous possibilities of protein production, enormous type of protein production from a same stretch of same stretch of the DNA. So you have a small same stretch of the DNA. From that same stretch of the DNA, different varieties. No, the RNA that is produced in the same single variety, but from that same single RNA, multiple varieties of RNA can be originated by modifying and editing the RNA, so that different varieties of proteins can be produced. So that increases the transcriptome in the cytosol at a given point of time. Okay, that's a very important role RNA editing plays. Now let's move to the next slide and discuss about the mechanism of RNA editing. Here we'll be talking about the mechanism of RNA editing, and I'll be looking at the mechanism for, in this case, this is about mechanism for modifying the existing nucleotide in the RNA during RNA editing. There are two separate mechanisms we'll be seeing. One is the modification of the existing nucleotides in the RNA, and the second one is insertion or deletion of nucleotides to the existing RNA. We are looking at RNA editing A to I, adenosine to inosine, how it is done. So recognition of RNA editing site is the step number 1. So in this case, we need to recognize the RNA editing site. So let's say this is the RNA, and let's say this is the site where the adenosine is found. This particular adenosine should be modified to inosine. So recognition is very important. And second step is formation of RNA editing complex. So all the proteins that are necessary to be converted. So this is the protein complex known as ADAR. Remember, because this is adenosine, so ADAR complex is needed. That is adenosine deaminase enzyme complex is needed, which will now stick to this particular location, which need to be edited. Okay, so here comes the whole complex ADAR locating itself to the RNA editing site, the site where A is present. Then comes the deamination of adenosine. The adenosine gets deaminated. Why and how it's deaminated? Basically, release of NH2, hydrolysis of NH2, and it's out. So this deamination will convert adenosine to inosine. So this is how the modification is done. Here we have inosine instead of adenosine. So inosine is now in place. The editing complex dissociation will be done. The editing complex still was present at this moment. Right now the editing complex will fall off, and finally the reading of the inosine. So when they read it during translation, they'll read inosine as guanosine. They'll read inosine as guanosine. So earlier it was A, but now it will read as G. So let's say if the sequence is something like this, it will now be read as this. Okay, so simple change of the reading frame, the way the nucleotide sequence is read during translation, and due to this change in nucleotide being read during translation, we have a different variety of proteins produced from the same stretch of RNA which was present earlier. So the same RNA, this is in unedited form, and this is the edited form at this end. This is edited. This is edited form of the RNA. Okay, that's how the RNA editing will be done, and this kind of editing is simple existing modification or change, modification of the nucleotide from A to I, or it can be done from C to U. Now, we'll also see another example. This is the example of RNA editing. We'll see the mechanism of RNA editing, but in this case it is simple insertion or deletion. Insertion or deletion of the RNA nucleotides. Step 1 is again recognition, the place which needs to be edited. Very important to understand which place needs to be edited. So this is again RNA. Let's say this is the place that needs to be edited. And let's say what we need to edit, there is a U. We need to cut this U out from that area. Alexa, stop. So this U need to be edited. This new U need to be cut out from this existing RNA. So how exactly we can do that? So again recognition by set of proteins out there, the enzymes involved there. Basically we know that there are RNA, guide RNA mediated process, how it is done sometimes. For the insertion process, the guide RNA plays an important role because the guide RNA contains extra nucleotide compared to the RNA that needs to be edited, and based on that extra nucleotide, it will recruit new nucleotide to the existing mRNA and insert the new nucleotide. But in this case, if you look at the deletion process in simple part here, is simply that the complex will be recruited here, the RNA editing complex. Okay, then comes the second stage, that is cleavage. We need to cleave this particular sequence in here, to cleave it, because in order to insert a nucleotide or delete a nucleotide, the existing strand must be cleaved. The strand gets cleaved out. Okay, so after cleaving out, the insertion or deletion can be done here. So let's say if there is insertion, so in this case, right after this there will be insertion. So insertion of, let's say, U, a U is inserted. Okay, so if we need to add another U here in this part, because let's say this is 5 prime, this is 3 prime, so obviously we need to attach uracil or any other nucleotide following the rule of polymerization, that is addition of a new nucleotide from 5 prime to 3 prime direction. So this addition will be done here, a U will be added. So what enzyme is involved for the addition? The enzyme is terminal uridilyl transferase. Terminal uridilyl transferase, also known as tutase in this case. Similarly, there are different enzymes depending upon the name of nucleotide which is being added or removed. We have different names. So in this case, uracil is added. So terminal uridilyl transferase is the enzyme that acts here and attaches nucleotide. Nucleotide, that is UTP, is needed in this particular process. So after the U is added, then there is a nick that is present in here, the nick to be sealed by ligase. So ligation is the fourth step. So ligation will be done, and then editing complex will release from this area. And after the editing is done, simply what we know is that U is attached or added here. Okay, earlier there was one U, but now next to that another U is present, the new U that is being added in here. Okay, so after this editing process is done, it's very important to have a quality control, a quality check whether this edited form of the mRNA is stable enough, whether this edited form of the mRNA is good enough to be translated into proteins or not. If there is any damage to this mRNA, if the RNA editing process does not continue with the proper manner and has any fault in the mRNA, then the mRNA will not be allowed to be translated into proteins. So that's why we have a quality control step, and after this quality control step, the RNA is translated into the proteins, and we have different variety of proteins because the mRNA that we have here, this is unedited form of the mRNA, and we have this is edited form. of the mRNA. So we have two separate forms of the mRNA, and both of these forms are responsible to produce different types of amino acid, different sets of amino acid. They'll produce different polypeptide chains. So a protein that is produced from here, let's say protein X, a protein produced from the edited form, let's say protein Y, and there will be different proteins X and Y. There will be totally different proteins, and that is the actual physiological role, that is the functional impact of RNA editing, is to increase the expression potential of the existing mRNA. So from the same existing mRNA, unedited form gives a different protein, edited form can give you different protein. Now, not always these edited forms give rise to a properly functional protein. The protein may not function well, the protein may not properly fold due to truncated version of protein production. That protein may not have a significant role, but still there is a chance factor that the cell will always take in order to produce different types of proteins, and once they produce different types of proteins, they are involved in the process of variety of protein production, and that is the main idea behind RNA editing. That's what the cells always gamble on the expression profile or expression pattern inside the cell. So that gamble will pay off. In many of the cases, the gamble pays off very well, and the cell receives variety of proteins to deal with, to have a different functions in the body. So that's all about the RNA editing process. The RNA editing process basics. What is RNA editing? When RNA editing is done, it is very important to know the location of all the different post-transcriptional modification, just like this RNA editing. So what is the location of RNA editing? We can simply say the location of RNA editing, it can be in nucleus or mitochondria. Nucleus and mitochondria, both are important locations. Nucleus, where particularly the modification of existing nucleotide, that is A to I, C to U, can be found in the nucleus, and also we find RNA editing in the mitochondria sometimes, not always, but sometimes the editing can also be found in the mitochondria. So these are the locations of RNA editing. Okay, so I believe you have a clear understanding of RNA editing now. If you get a clear understanding, please hit the like button, share this video with your friends and colleagues, and subscribe to this channel to get more videos like that in future. And watch all the different lectures of this series regarding the RNA modification, the post-transcriptional RNA modification, including the RNA splicing, 5' capping, 3' polyadenylation, and RNA editing. Thank you everyone. Bye.